Welcome student on the third online class on plant growth and development. So today we are going to start the new concept called plant development. Before that, in the redifferentiation process, they asked some question on page number 245 NCERT. That is the second paragraph. List the sum of the woody dicotyledonous plants that are the products of redifferentiation. What are the tissues in the plants that are the product of redifferentiation products? Take the answer for that. Secondary xylem, secondary phloem, cork, secondary cortex, cork cells. These are the example for redifferentiated tissues in the plant cells, woody dicot plants, which are the redifferentiated tissues. Secondary xylem, secondary phloem, secondary cortex, cork cells. These are redifferentiated tissues. Another question they have asked, what would you call parenchyma cells that made to divide under laboratory condition during plant tissue culture? That is the question. That process is called dedifferentiation. Under laboratory condition, permanent cell divide to form new tissue or meristematic tissue called dedifferentiation. These are the two the questions which are given in the dedifferentiation process. Okay. Now we are going to start the new concept here. What is the name of that concept? Is called plant development. What is the plant development process? So what is the plant development process? Process includes all the changes in the life cycle of organism from seed germination to the senescence. It is a life cycle in the life cycle of organism from seed germination up to the senescence. That means the death of the plants. Up to that, whatever the changes takes place in the organism, that changes are called development. Say for example, this is a zygote. From zygote, 16-celled embryo, then heart-shaped embryo. Then remember, there are the juvenile stage of the plant. Then with the root and shoot formation process takes place in the plants. Then the mature plant formation, that is, this is the adult stage of the plant, this is the juvenile stage of the plant, and this is the reproductive stage of the plant. So these are the various developmental processes in the plants that we have to learn under the concept called development. What is this development? It is a sum of growth and differentiation. Development is sum of growth and differentiation. Sum of growth and differentiation we can call development. It is represented as follows. How it is represented? That chart we have to study here. So this is the development of higher plant. This diagram is showing development of higher plant from the zygote. From zygote to 16-celled stage embryo, heart-shaped embryo will form. Then juvenile plant. Then juvenile plant gives rise to adult plant, which is having vegetative parts only. And this is the reproductive plant where the flowers are present on the plants. Later that flowers are converted into these fruits. And fruits contains what? Seeds. So seeds give rise to again new plant. So these are the events which are taking place in the plant development from the single cell zygote, which is formed in the zygote is going to be formed in which sac? Embryo sac. Okay. Now what is exactly development process? Diagrammatic represents sequence of events. These are development of higher plant, mature plant, how it is going to be developed. That is diagrammatic event they have shown here. The meristematic cell undergo plasmatic growth. Plasmatic growth means cytoplasmic growth and differentiation process. During plasmatic growth, what happens? This is the meristematic cells. These are the meristematic cells. So these meristematic cells will undergo elongation, that means vacuolization, protoplasmic division. Content of protoplasm will get increased. If content of protoplasm and vacuolation happens, so vacuolation and content of protoplasm increase, that is plasmatic growth. Plasmatic growth means in meristematic tissue or in meristematic cells. These are the meristematic cells where protoplasm content increased, protoplasm increase, and vacuole increase. This is called plasmatic growth. And after that, what will happen? This elongated cell undergo maturation. Maturation means differentiation process only. Maturation means what? This elongated cell will acquire permanent definite size and shape. Cell wall become a thick cell wall. Okay, cell wall thick. Nucleus, vacuole is there. Nucleus, vacuole is there. So here the permanent size and shape cell will acquire, and the differentiation means after the elongation cell will undergo maturation. This is a mature cell. After elongation the mature cell will be formed, and after the age of the death or the age of the cell will get completed, that aging of the cell is called senescence. Aging of the cell is called senescence. Senescence process we can call it is. And after the senescence the cell will undergo death. So cell death process we can call it is. So this is what the life cycle of this is some of the process called development process. In development process, the meristematic cell initially their cell will undergo the elongation because inside the vacuole will get increased, protoplasm content will get increased. After that elongated cell become mature because their cell wall become thick, and cell wall acquiring thick position, then the cell will undergo at one point aging process. That aging process is called senescence. After that cell death happens and life cycle is completed. Okay, like that. The plasmatic growth, see here, sometime differentiated cell again acquires cell division ability, again forming meristematic tissue. After the differentiation, you know, this is a permanent tissue is formed. What is the permanent tissue is formed? Parenchyma. See, after differentiation, this permanent tissue again regain the ability of cell division, again it will form meristematic tissue. What is the process we can say? I already described about this, the differentiation process. Okay, so this is what the process called development diagrammatic representation. Development of cell of higher plant cell development into what we can say particular organ, or after death what will happen. Again that some of the cells will get acquired cell division ability, again meristematic or differentiation process. So this is the diagrammatic representation of development process. That's why development is nothing but growth and differentiation. See here, plasmatic growth means vacuole and cytoplasm growth will be happens. Differentiation means a maturation, that means cell become mature, permanent, and cell wall thick it is acquired, that is called differentiation, referred as maturation process. Okay, now so plants follow different pathway in response to environment or phases of life to different kinds of structure it will form. That means plant follow different pathway in response to environment or phases of life to form different kinds of structures. The plants have ability. This ability is called plasticity. In response to different environmental conditions, plant can form different structures in their leaf. Their structure of the leaf also will get changes. Shape of the leaf also get changes according to the environment. that in response to environment, remember, the structures or plants follow different pathways in response to environment or phases to different kinds of structure to form different kinds of structure. This ability is called plasticity. Means what? Say for example, heterophily due to phases of life. In response to plant, heterophily in the plants means what? Different kinds of leaves in the single plant only in two different phases of life. For example, this is a cotton plant. In cotton plant or coriander plant, see here Larkspur plant is given. A diagram is a Larkspur. In cotton, in coriander and Larkspur plant, heterophily due to phases of life. That means what? Heterophily means occurrence of different types of leaves. Hetero means different types of leaves in two different phases of life. What are the phases of the plant life? Juvenile phase and adult phase. In juvenile plant, how is the heterophily in Larkspur plant? The size of the leaf will be very small. And same plant at the adult stage, see the leaf becomes a trifoliate like this. So this is in adult, the size of the leaf will get changes. How much size of the leaf will get changed, you can observe here. It means that in juvenile stage and in adult stage, shape of the leaf will change. That means heterophily due to phases of life. Plant having two phases of life. Juvenile phase and adult phase and reproductive phase. Three phases generally there are. Juvenile phase means small phase, vegetative phase, and adult phase, and then reproductive phase means which is having flowers. So here taken only two phases, juvenile and adult phase. Observe here the shape of the leaves in the juvenile stage and the shape of the leaves in the adult stage. Totally different. This is called heterophily due to phases of life. Due to phases of what? Life. And next, heterophily due to environment. Heterophily due to environment means what? See here example they have given buttercup plant. In the buttercup plant, B diagram, see in the terrestrial habitat, how is the shape of the leaf? And when water present in the, plant present in the water, see the shape of the leaf. Totally changes. That means in different environment, in water environment, on land environment, shape of the leaf is totally different. And that to be on the water, in the water, shape of the leaf completely different. That means shape of the leaf depends on the environment where it is present, depends on the habitat where the plant is present. Suppose the plant present in the on the land, the shape of the leaf like this trifoliate. And when it is present in the water, see it is a scaly leaf-like structure will be appears. This is observed in case of plant called buttercup. Different types of shapes of leaves in different environments. That is a heterophily due to environment and heterophily due to phases of life. Juvenile phase different shape, adult phase different shape. That is the heterophily. That is generally observed in a plant called cotton, coriander, Larkspur plant. And buttercup plant, heterophily due to environment we have observed here. So this is nothing but we have studied about shapes of leaves in response to environment or phases of life. Different kinds of shapes of leaves will be formed in the plant. Hope students you understood about what is development. Next, Some of the factors controlling plant development. Which are the factors controlling plant development? External factors and internal factors. Which are intrinsic factor, intracellular factors. That is also very important role play for the plant development. What is intracellular factor? DNA, RNA, such a factors plays very important role during plant growth and development. And that is nothing but genetic factor, which are those that RNA, DNA. These are the genetic factors responsible for intracellular growth, because what kind of DNA is there, that kind of protein is formed in the plant, and that protein it is essential for the plant growth. That is inside the cell. These are the factors. Intercellular factors, that means nothing but plant growth regulators. Which are the plant growth regulators? Like auxin, like cytokinins, like gibberellins. So these factors plays a very important role during plant growth. So this is helps for shoot formation, this is helps for root. You know the function of every hormone function we have to study in the next point. Auxin function, cytokinin function, gibberellin function. These are the plant hormones. They are also responsible for the plant growth. How? Every hormone, each and every hormone we have to study in detail. Their role in the plant. What is the role in the plant development, that we have to study. Okay. Extrinsic factor, which is that light is essential, temperature is essential, water is essential, oxygen is essential, nutrition is essential. These all are the factors are also required for the plant development. You know, some of the growth and differentiation is nothing but plant development. Remember, without photosynthesis there is no growth. If growth is not there, there is no differentiation. And for photosynthesis or for respiration, there is a proper temperature. Optimum temperature should be very essential. For photosynthesis, there is a proper providing of water. Raw material is very important role it will play. As well as oxygen for the respiration process. You know that C6H12O6. In that, what is combined? So oxygen gives rise to 6CO2 plus 6H2O. Along with how many molecules of ATP? 38 molecules of ATP. That is also needed for the plant growth. Along with that nutrition, that means you know that the mineral nutrition topic we have studied. There are two nutrients which will be essential for the plant growth. Which are the nutrients? Micronutrients and macronutrients. These are also playing very important role during the plant growth and development process. Without light, there is no photosynthesis. No photosynthesis, remember, there is no food production. And all photosynthesis and respiration reaction, if plants has to be happen in the optimum temperature, is very essential, because the photosynthesis reaction or respiration reaction all are enzymatic reaction. Photosynthesis and respiration reaction, how reaction? Those are enzymatic reactions, and for proper enzyme work, optimum temperature is essential. And for photosynthesis, what is the raw material is required? Water is the raw material is required. And remember, during differentiation, there is a vacuolization happens. Protoplasmic content of the cell will get increased. Vacuole size will get increased. For vacuolization, what is essential? Water is also essential. For protoplasm formation, what is required? Nutrients also required. So remember, these all are the factors which are responsible for plant development. Plant development. Okay. Okay, now we have to study plant growth regulators. That is intercellular factors, as I said, auxin, cytokinin, gibberellin, like that. So these factors are small, simple molecule that regulate the plant growth. These factors are very small, simple molecule, and that regulate the plant growth. Okay, based on, and they are having diverse chemical composition. They are having diverse chemical composition. Based on function, plant growth regulators are classified into two types. Which are the two types of plant growth regulators? One is called plant growth promoters, other is called plant growth inhibitors. Promoters means what? They help for cell division, cell enlargement, trophic growth of the plant, pattern of formation, flowering, fruiting, seed formation. These all are growth promoting activities. What is that? Cell division. Number of cells will increase. That means growth promoting activity. Cell length increase, cell enlargement. That is a growth promoting activity. Trophic growth. That means root growing towards the water. That is a trophic growth. Wow. That is called hydrotrophic growth. Stem growing towards the light. That is called as phototrophic growth. So that is called trophic growth. For trophic growth, these regulators are responsible. Pattern of formation. Pattern of formation means leaves pattern of formation, flower pattern of formation. So for that, flowering process, fruiting process, and seed formation process. All these are growth promoting activities. So for growth promoting activities, growth promoter hormones are essential. Which are those? Auxins, gibberellins, cytokines. These are helps for growth promoting activities. Growth inhibiting activity means growth stopping activities like seed dormancy. Seed dormancy means seed never germinates. Seed will undergo resting period. That is called seed dormancy. And what is abscission? Abscission means falling of leaves. Falling of leaves, flowers, fruits, etc. That is called abscission. Abscission. So seed dormancy means resting period. Seed never grows, seed never germinate. That means growth stop, growth inhibiting. Leaves falling, flowers forming, falling, fruit falling process means abscission. Abscission, leaves falling process, fruit falling process, seed falling process. So falling processes are called abscission process. Those are called growth inhibitors. Growth inhibiting processes. Seed never germinates means seed never form a plant. That means growth is inhibited here. Leaves never become a larger. Before larger, sometime premature falling of leaves happen. Premature leaf fall happen. This premature leaf fall happen. That process is called abscission. See, for example, respond to own stresses to abiotic and biotic and abiotic origin. Abscisic acid and ethylene plays very important role. That means if suppose plant is having any wound or there is a water stress, there is any biotic agent is less, or there is a abiotic stress, that means water stress, sunlight stress, that means sunlight is less there, that is abiotic origin. Some living organism, pollinator is not there, that is a biotic stress. If pollinator absent, abiotic origin, certain factors like water absent, pollinator absent, that time plant growth inhibitors are more there. Which are the more plant growth inhibitors? Abscisic acid and ethylene. Ethylene is fitting into the both groups, inhibitor as well as promoter, but largely inhibitors there. Are you understood? Hope you understood, students. That is what is the inhibitor process. Inhibitor means growth stopping activity is called growth inhibition. Seed resting period, seed never form new plant, that is a growth stopping activity. Leaves falling process, leaf fall happen, premature leaf fall, that is the abscission process, that is because of the inhibitors. Which inhibitors are produced? Abscisic acid and ethylene. Inhibitors are also produced in response to, if suppose plant is having any wound. If plant is having any wound means it is having any injury. Suppose it is having any injury, or if plant is having waterless, or the plant is have pollinatorless. Pollinatorless means biotic stress. That is a biotic stress. And the waterless means abiotic stress. If such are the things are there, then remember what hormones are produced? Plant growth inhibitors are produced. And for cell division, cell enlargement, trophic movement in the plant, trophic growth means what? Hydrotrophic growth, root growing towards the water, that is hydrotrophic growth. Stem growing towards the light, that is a phototropic growth, like that. Those are called trophic growth. Pattern of leaves formation, flowering, fruiting, and seed formation. That time plant growth promoters are produced, and seed dormancy and abscission, plant growth inhibitors are produced. Okay. Now, first hormone we have to learn. What is the name of that hormone? Is called auxin. Before going auxin, we have to divide the PGR broadly into different types. That we have to study. Before going to the auxin, we have to study in detail points whatever given in the NCERT textbook. Concentrate. Okay, which are those? Indole compounds. Indole compounds. That is indole 3 acetic acid. The hormone name is, that is in short form, you can going to write here IAA. Adenine derivative. This is a chemical compound of indole hormone. It is indole compounds. Adenine derivative. Adenine derivative are n raised to 6 perfurine aminopurine. Aminopurine, which is commonly called kinetin hormone. Kinetin hormone. Remember, these are the auxins, natural auxin hormones. These are the cytokinin hormones. Cytokinin hormones. Chemical name of this cytokinin hormone like this. Kinetin. These are adenine derivative. Kinetin hormones are called adenine derivative, and indole compounds are auxin compounds, naturally occurring auxin compounds. Okay, now carotenoid derivative. Carotenoid derivative. Which are the carotenoid derivative? ABA, abscisic acid, is a carotenoid derivative compound hormone. Terpenes, chemically terpenes these are. Which are the terpene compounds? Gibberellic acid. Gibberellic acids. These are the terpene compounds. We can call commonly GA3, gibberellic acid, which is more common. And gaseous hormones. Which are the gaseous hormones? That is nothing but ethylene, C2H4. So these are called plant growth hormones. These are variously described plant growth hormones. Indole compounds are auxins. Cytokinins are adenine derivative. Abscisic acids are carotenoid derivative. Gibberellic acids are terpene derivative, and gaseous hormone ethylene. These are chemical derivatives or chemical present in the hormone. Chemicals present in hormone. Plant hormones. Okay, auxins are AIA, indole acetic acid, auxin is present. Okay, so these are the different kinds of chemical derivatives of hormones we have studied, and we have studied also the various plant growth hormones. Some of that gaseous hormone is, which is that ethylene. Ethylene is the gaseous hormone. So here plant growth regulators divided into two groups that I also explained you, that is plant growth promoters and plant growth inhibitors. Promoter means which is promoting the growth of the plant. That means cell division, cell size increase, trophic movement in the plants, fruit formation, seed formation, flower formation. These all are promoting activity. And seed dormancy means seed never produce new plant. That is a resting period of seed. And leaf falling process, abscission. These are growth stopping activities called growth inhibitors process, we can say. Okay. The first hormone we are going to study. What is the name of that? The auxins. Auxins hormone, how it is discovered by? Discovered by scientist Charles Darwin and his son. Francis Darwin observed the coleoptile of canary grass responded to the unilateral illumination by growing towards the light source, and it was concluded that root tip of coleoptile caused bending of the entire coleoptile. Tip of the coleoptile caused the bending of entire coleoptile. Do you know coleoptile means what? How auxins was discovered? Auxin in Greek means, Greek meaning of this auxin refers to grow, means it is a promoting hormone, growth promoting hormone. See, what is a coleoptile? First we have to understand. You know, this is the grass embryo we have studied in the maize embryo plant, maize seed embryo, and in that we have studied in the maize plant seed, maize seed. This total maximum part is called endosperm. And this is the particular area is called embryo. Embryo is having two parts, plumule and radicle. Remember, this is a seed section of monocot. In monocot seed, we are having two parts. Larger part is endosperm and minimum part is the embryo. That embryo is made up of two parts, plumule and radicle. Plumule and what? Radicle. On plumule, there is a cover called coleoptile. And on the radicle, there is a cover called coleorhiza. So we have studied this one. On radicle there is a cover called coleorhiza. On plumule there is a cover called coleoptile. Okay. Now Darwin, remember, from the plumule shoot is formed, and this is experiment on the shoot experiment. That means on the shoot, see, on the above the plumule there is a coleoptile. That means at the tip of the shoot there is a presence of coleoptile. This is a coleoptile. Remember, on the plumule in the monocot seed, coleoptile is present. That means plumule gives rise to shoot, and shoot tip is having coleoptile. So that is what we have to study. Coleoptile demonstrates tip of the coleoptile is the source of oxygen. How source of oxygen to the source of oxygen? How the coleoptile will respond? See, Charles Darwin and his son Francis Darwin. Charles Darwin and his son Francis Darwin, they observed that in the coleoptile of canary grass, this is the canary grass coleoptile tip. This is the coleoptile tip in the canary grass. When illuminated to the unilateral light, canary grass illuminated to unilateral light, what will happen? This coleoptile growing towards the light source, it will bend. See, this entire coleoptile, that means tip of the shoot. This is the shoot. This shoot tip having coleoptile. This coleoptile, when illuminated to unilateral light, it is bending towards the light. So tip of this coleoptile bending towards the light. So why it is bending towards the light? So F.W. Wendt scientist from the tip of the coleoptile of oat seedling, the oat seedling plant, from this tip of the coleoptile, he isolated chemical. What is the chemical he isolated from tip of the coleoptile? Oxygen. Because of the oxygen source, because of the oxygen in canary grass, that bending of coleoptile happens towards the source of light. At that time, this bended tip from that coleoptile tip, F.W. Wendt, scientist, isolated oxygen hormone. So series of experiment. Remember here, this line, this coleoptile tip is growing towards the light. That means they are showing the phototropism. For phototropism, remember, for phototropism means growing towards the light. For phototropism, which hormone playing an important role? Oxygen hormone playing an important role for tropic movements. That means growing this coleoptile tip towards the light, bending towards the light. This bending is caused by oxygen hormone here. Because of oxygen, this coleoptile tip is bending towards the light. Later they isolated the hormone from that. What is the name? Oxygen. Oxygen also first isolated from, oxygen was first isolated from human urine. Coleoptile tip also F.W. Wendt isolated, but also that is isolated from human urine also. And oxygens are produced by growing apices of the stem and root, from where they migrate to the different regions of their action. Growing region of tip and tip of the shoot and root, what is going to be isolated, what is going to be produced? Oxygen is going to be produced there. The growing tip of the shoot, growing tip of the root. So shoot tip, as well as root tip, where oxygen is going to be produced. And from there they produce here and migrate to deep, where they are going to be make action. At that place they will migrate. They produce at the tip region and they migrate for their action region. Okay, this is the diagram which is showing the, and remember this is he isolated F.W. Wendt oxygen from oat seedling plants. Oat seedling plants he isolated oxygen first time. Okay. So this is about the hormone called as oxygen discovery we have studied. Oxygen also discovered from the human urine. First time discovered from human urine. They produce at one point and go to the migrate to the other place where they will do their action. Suppose they will produce at the root tip, they will go for the shoot for their action, or produce their shoot tip, they action their function on the root tip. That means at which place the oxygen produced, not that place it will act. It will act on the other place where it is having a function. This is a discovery of auxin hormone by Charles Darwin and his son Francis Darwin. So first we can see the types of auxin first detail. Later paragraph on the page number 248, they have given discovery of cytokinin, discovery of gibberellins and discovery of ethylene they have given. First I will complete the auxin discovery, auxin role in the plants and auxin types we can discover first, we can study first, and then we can go for the again cytokinin discovery, gibberellin discovery, ethylene discovery, that we can study here. First, what are the types of auxins? Number one, natural auxin, synthetic auxin. Natural auxin and synthetic auxins. Which are the natural auxins? Indole 3 acetic acid. These are called natural auxins. Indole 3 butyric acid. Natural auxins. They are isolated from the plants. Synthetic auxin, Na, naphthalene acetic acid, 2,4-D, dichlorophenoxy acetic acid. These are called synthetic auxins. See, they have given the diagram also. IA, that is natural auxin, IA, IPA, that is a natural auxin, and synthetic auxin, dichlorophenoxy acetic acid, naphthalene acetic acid. This is the structure of naphthalene acetic acid. This is the structure of dichlorophenoxy acetic acid. Dichloro 2,4 dichlorophenoxy acetic acid. There are two chlorine atoms, dichlorophenoxy, phenyl group is there. Acetic acid group is present here. Dichlorophenoxy acetic acid. 2,4. 2 means carbon number 2 and carbon number 4. Chlorine atom is present. 2,4 dichlorophenoxy acetic acid. Naphthalene acetic acid. So here naphthalene is there. Naphthalene structure and acetic acid is present. And indole butyric acid. Indole, this is the indole group, and butyric acid is present here. As well as indole acetic acid. This is the indole group and acetic acid group is attached. So this is what these are the chemical structures of different types of auxins hormones. What are the functions of auxin? Number one, to initiate the rooting in the stem cuttings for plant propagation. If you have taken the stem cut like this, stem cut we have taken, and in this stem cut, there should be rooting. Rooting means root formation process. For that, what is going to be added? Hormones, auxin is essential for that rooting in the plants. So, for rooting as well as flowering in pineapple, fruit and leaf drop at early stages. Okay. Concentrate here. Rooting, auxin function or physiological roles in the plants. Suppose this is a sugar cane we have taken. Sugar cane stem cuts we have taken like this. These are the stem cuts of sugar cane. We can call it stem cuttings. When we grow these stem cuttings into the soil, so this stem cutting is having bud like this. Bud is there, and from this bud there will be formation of roots. So rooting in the stem cutting, rooting in stem cutting by auxin hormone, particularly which hormone is applied? IA hormone is going to be applied. So in that stem cutting we get the roots, particularly in the sugar cane. Okay. Now this is the function of auxin hormone. pineapple plant. In pineapple plant, flowering process is promoted by auxin hormone. Okay. To prevent the fruit and leaf drop at early stages. At early stages, leaf drop, fruit drop is prevented by, this is the auxin hormone. Rooting in the stem cut, suppose this is a stem cut, in that rooting process, if you add auxin here, rooting happens here, root formation takes place, and even it is promote flowering in various pineapple plant. Fruit and leaf drop, this leaf drop and fruit drop in early stages is prevented by auxin hormone. Promote abscission in older leaves and fruits. When leaf become old, that will fall down. Old leaf falling process is promotion is carried out by, old leaf falling process means abscission in older leaves means old leaf falling. Old fruit falling process means abscission in the old fruit falling process. That hormone, auxin hormone, helps for older leaves and older fruit falling process, that is abscission process. Induce parthenocarpy. What is parthenocarpy? That is fruit formation without fertilization. Fruit formation without fertilization in tomato, it will induce parthenocarpic tomato formation. Artificial parthenocarpy we can call it is. Artificial parthenocarpy. Seedless tomato formation process will be takes place. Okay, next. They are also used as a herbicides. Herbicides means a weed killing process. Weeds. Unwanted plants in the crops is killed by using these hormones, auxin hormone. Particularly 2,4-D is used to kill dicot weeds. It does not affect the monocot plants. In monocot plants, dicot weeds are killed. Say for example, maize crop is there. In maize crop land, dicot weeds are killed by using 2,4-D plants. So what are weeds? Weeds mean unwanted plants. Unwanted plants are killed by. Now unwanted plants are, which are those? Unwanted plants are dicot. And crop plants, which are those? Monocot. Suppose we are having agriculture and you have planted the maize plant there. Maize plants are monocot. In that unwanted plant, dicot plants are going. We have to spray the herbicide 2,4-D on dicot plant, that is unwanted plants. That does not affect our maize plant or the main crop. So it is killing the unwanted dicot plants from monocot agriculture plants. So that is the function of the 2,4-D, which is used to kill the harmful or unwanted plants. Controls xylem differentiation and helps in cell division. It is also helping in xylem differentiation and cell division process. Cell division process and xylem differentiation process, this hormone is helping. In higher plants, one of the role of the auxin is apical dominance. Apical dominance means what? Generally, growing of apical bud inhibit the growth of lateral buds. It is known as apical dominance. Means what? Auxin induces apical dominance. Means what? Suppose this is a plant. We are having a node, internode and tip region is there. If a tip is growing faster, older leaves, younger leaves like this. If tip is growing, side buds are not growing. Axillary bud, growth, suppress. This axillary bud is not giving branches, its growth is suppressed or stops. Because which bud is dominant? Apical bud is growing. If apical bud is growing and lateral bud growth is suppressed, then this phenomena is called apical dominance. That means tip part growth maximum and axillary bud growth suppress or stops. That kind of phenomena is called apical dominance, because tip is growing and lateral bud growth stops. So now if you break this apical dominance by process called, you can break this apical dominance by decapitation. Decapitation means what? Decapitation means breaking of apical dominance. So why apical dominance happens? Because at the tip, which hormone is playing important role? Auxin hormone. Because of the auxin hormone, tip is growing maximum and side buds are not growing. That is apical dominance. Now we are breaking apical dominance means cutting of this tip. When you cut the tip of the plant, tip is cutted like this. After cutting of tip, this axillary bud growth happens and terminal bud growth suppress. Axillary bud growth happens, terminal bud growth suppress. Tip growth stops. Tip growth stops and axillary bud growth. This process is called breaking of apical dominance or decapitation. If you remove this shoot tip, the hormone auxin goes to the axillary bud and axillary bud growth happens and tip growth will stop here. This tip growth is never happens. This tip growth will stop here and axillary bud start growing. These are the axillary bud start growing. So apical dominance we have break here. Breaking is called decapitation process. So apical dominance is induced by which hormone? Auxin hormone. And that is removed. When you remove the tip, the auxin goes into the axillary bud and axillary bud will develop and form a shoot system. This is called the apical dominance and making process. Apical dominance, generally decapitation, lateral bud goes. It is widely applicable in which plants? Decapitation is applicable in tea plants. Tip of the plants is going to be cutted there. This process is called, this is a hedge making process. Hedge making. In tea plant, you have observed that workers are cutting the tips of the tea plant. If they are cutting the tips of the tea plant, maximum leaves will be appears from the axillary parts. Or axillary parts again shoot formation takes place and the leaves production maximum. If leaves production maximum, tea product will be maximum. In tea plants, such a decapitation process is going to be carried out. That is the process is called hedge making process. See, observe here. These are the lateral buds. Lateral buds here we have cutted this apical tip. We have cutted. After cutting tip, we are having this axillary bud forming new shoots here. So apical bud is removed here. Apical bud is removed. After removal of apical bud, axillary bud, that is a lateral branches. Here we are having axillary bud, and from this axillary bud, lateral branches are produced. From this axillary bud, what branches are produced? Lateral branches are produced, and the maximum leaves are produced from the plant. Okay, this process, removal of shoot tip, is called decapitation process, which is Generally in tea plantation process they will going to carry out, or we can call hedge making process. Okay, this is the first hormone we have studied. What is the name of the hormone? Is called auxin. Let me have a revision of auxin hormone. Auxin was discovered by scientist Charles Darwin and his son Francis Darwin. They, in oat seedling, they illuminated the polyptile towards the source of light. The bending of polyptile happens. From that bending, FW went isolated auxin hormone. It is also isolated from human urine also. Okay, that means first time it was isolated from human urine. So the auxins we have studied, and generally they are produced at the growing tip of the root and stem, from where they migrate to the other region of their action. Generally shoot tip and root tip, what are produced? Auxins are produced, and they migrate to their, where they will going to do their action, where they will going to perform their action, that particular area auxins will migrate. If produced at the shoot tip, migrate towards the root for their action, or produced at the root tip, migrate towards the shoot for their action. So for this is the example what I am giving you for the auxin role. So I said the auxins are classified, the two types. So I said natural auxin and synthetic auxins. Which are those natural auxins? Generally we have seen those are generally produced in growing apices of stem and root. Auxins, natural auxins are IA and IBA. Those are natural auxins, and synthetic auxins are generally Na and 2,4-D are called synthetic auxins, and their chemical structure also I described. And what are their roles that we have also described? Flowering and fruit leaf drop in early stages, that is prevented. Early stages, if fruit fall, leaf fall, what will happen? The productivity of the plant will get decreased. So it is used to kill the dicot weeds among the monocot plants. That is also herbicide use, that is unwanted plant killing process. Xylem differentiation process, cell division process, for all these processes that having role. So this is most important function we have studied, apical dominance. Because of the tip region is growing more because of the presence of auxin. And axillary bud will dormant. When you cut this tip, axillary bud will grow. That process is called decapitation process we have studied. Okay. Okay. Next hormone. What is the hormone name? Is gibberellins. That hormone we can study in the next video. And I said you that parthenocarpy, particularly in tomato, fruit formation without fertilization, seedless tomato formation, the auxin role is having. Okay. So next class I will going to describe you, or I will going to explain you hormone called auxins, gibberellins and cytokinins. Okay. Thank you. All the points from NCERT I am covering, and some of the extra things also I will try to give you. Okay, please concentrate and please see all the videos properly. Thank you.